For a PTC tasting lab, it's important that you know some key background information before attempting to write your CER. So just a quick review of the article you were supposed to read about PTC. PTC is actually was accidentally discovered in 1931 by a chemist who was doing an experiment on something completely different, and noticed that when this powder accidentally blew into the air, that some of the scientists nearby could taste it, but that others couldn't. So he was intrigued by this. So it led to eventually down the line in 2003, the discovery of the PTC gene. Okay. This here is a picture of PTC paper. And that is the paper that our in-class students use in order to collect some data. Now we classify people as being either tasters or non-tasters. On average, 75% of people are able to taste PTC, while 25% cannot. This PTC gene is linked to being able to taste bitter flavors such as dark chocolate and coffee. A little bit about the anatomy behind the tasting of the PTC gene. Okay, first of all, this gene, known as TAS2R38, was discovered in 2003. Okay, and they found that this gene affects the receptor shape on the tongue. Okay, so if you follow this diagram down, talking about the taste buds and these different types of cells and the nerves that carry the signals, we end up down here at the bitter taste receptors. The tip of each one of these important cells has this, they're called gustatory cells, has these bitter taste receptors, and they can detect a wide variety of different compounds. Okay, if these receptors are stimulated, then a message is sent to the brain, letting the brain know that it is bitter. But how it works is, in this signal transduction pathway, what happens is the gene affects the shape of these receptors. Okay, so if you actually have the gene that enables you to taste the bitter, then your receptors are what we would consider normal shape, and PTC binds to your receptors pretty easily, and the message is sent quickly to your brain. If you lack this gene, then the shape of these receptors has been altered, much like what happened to like an enzyme that's being denatured, and therefore the signal is not received and never gets sent. To link this a little bit to natural selection, PTC is not necessarily a chemical that's found in nature, but it does strongly correlate to an organism's ability to taste bitter substances. And in the wild, many plants will produce a toxic compound in order to prevent herbivores from wanting to eat them. This toxic compound usually has a bitter taste. So what we found is, in terms of evolution, it's evolutionary importance that organisms can taste bitter, because then it sends them a signal not to eat that plant, because the plant has some types of toxins that are being produced inside of it. This is what PTC actually stands for. All right, it's also known as phenolphthaleurea. Okay, and it is very substance like it physically resembles some toxic alkaloids that are found in poisonous plants. Now, to taste or not to taste, scientists do classify the ability to taste this type of bitter as being complete dominance, but there are different degrees of tasters. It is not just this one gene that enables you to taste bitter substances. They believe it's actually 30 different genes. But we're just going to focus on the PTC one. Okay, it is inherited as a dominant trait. And when you can see here, when you're looking at the PTC and the receptor, if you're a homozygous dominant, you can see how the PTC can bind to the receptor and the message is sent inside, letting your brain know you're tasting something bitter. If you're homozygous recessive, then your receptors are misshapen. The PTC does not bind, therefore you don't taste. Okay, and then they found that kids having both tasting and non-tasting receptors, heterozygous, so tasting is dominant, you're actually able to taste the bitterness. Sometimes scientists actually believe that could be an evolutionary advantage to be heterozygote, because they think your non-tasting gene also gives you the ability to taste different, even more unique substances. Okay, a little bit about potential health risks and applications. Studies do find that strong tasters are less likely to be smokers because smoking has like a bitter taste to it. Might not enjoy as bitter food as much as non-tasters like broccoli or dark chocolate. It does show that over time, these receptors can change or kind of like enzymes kind of become denatured, so you're less likely to taste things. What we did in this lab is students that were in person were given three different things to taste. First, they were given a strip of PTC, and they took this strip and they touched it to their tongue. So it looks like these strips right here at the beginning. Okay. And then if they could taste PTC, they could taste that bitter flavor, then they checked yes. Then they were given a strip called sodium benzoate. Same thing. This is just a different chemical. Okay, kind of has an irony taste, and they were to check yes or no for being tasters. And then finally, they were given a strip that contained a molecule called thiourea. Again, testing for the presence of that gene, the ability to taste thiourea. And they checked yes or no. What we are then going to eventually do with this information on Friday is you are going to perform a chi-square analysis where you are going to, based off the data here, where we are expected to get 75% of people that can taste and 25 that cannot. So that's our expected values. We're going to determine our observed values of PTC. How many tasters and non-tasters did we have in class? You're going to do a chi-square analysis to see if our AP biology students that were in class is a direct reflection of the general population. So in other words, are our percent tasters close to the human population's percent of tasters?